Hi, my name is Robin Amara Hurd, and I'm here today to talk to you about micro and nanoplastics and whether they are an environmental risk for cancer. The Center for Disease Control defines cancer as a disease in which abnormal cells divide out of control and can invade other tissues. Essentially, they've made a home where someone else has already been living. Environmental, genetic, and lifestyle factors influence our risk for developing cancer. That means that what we eat, our daily habits, and even our living environment may contribute to our susceptibility to cancer. Plastics have been a revolutionary invention. They've helped to make items affordable, lightweight, sanitary, attractive, and durable. Most of our food is either processed in factory farms or picked by hardworking people and then transported. What you might not know is before the food gets to us, plastic is used in the agricultural industry to help keep soil moist and to help with timed fertilizers. That's one way plastic is making its way into our food. Imagine 10 average-sized refrigerators. Altogether, that weighs about 1 ton. The latest data states that in 2018, U.S. plastic production reached 35.7 million tons. That's a lot of refrigerators. This map shows a pathway of plastic as it makes its way through our environment. When plastic and chemicals that are used to make isn't disposed of properly, it can end up in places like rivers, oceans, and landfills, and into our food, especially seafood. And it takes a really long time for them to break down. And even then, microplastics are defined as synthetic solid particles or polymeric matrices with regular or irregular shape and with size ranging from 1 nanometer to 5 millimeters of either primary or secondary manufacturing origin, which are insoluble in water. Nanoplastics are even smaller and less researched, but could potentially be more easily absorbed in cells and tissues. They are smaller than 1 nanometer in size. Currently, research on plastic and cancer risk discuss either the physical plastics or the chemicals used in the manufacturing. Microplastics and nanoplastics also have the ability to absorb heavy metals and forever chemicals. The CDC found in 2004 that detectable levels of chemicals like BPA were found in 93% of 2,517 urine samples from people 6 years and older. And it's not just there. Microplastics and nanoplastics have been found in the intestines of seafood and rice roots. Let's get into the research to find out if micro and nanoplastics are an emerging risk for cancer. An in vivo study by Sulukan et al. Looked at the effects of nano-sized particles of polystyrene in the offspring of zebrafish. In vivo means that the research was conducted on a living organism. This figure represents the methods of the study. First, the scientists injected some zebrafish with the polystyrene particles and then created a control group of the zebrafish that were not injected. The offspring of the injected zebrafish were then raised in an environment without exposure to plastic to see if their parents' exposure affected their development as embryos. What is found in this study? The offspring of the zebrafish that were injected with plastic had changes in their cell growth, cell metabolism, and also had inflammation. These changes are represented in this figure from the study. The first part of the figure displays green staining. The staining is from a specific dye that only attaches to the DNA of dead cells. So what you see indicates the accumulation of dead cells in these zebrafish. Dead cells are found in fish that are unable to get rid of unneeded cells through a process known as cell death. Cell death is essentially the body's way of removing cells that are either too old or damaged. You can see how much more intense the dye is on the fishes that were born from parents injected with plastic, which is what we will call the experimental group. Next, we can see the concentration of free radicals or reactive oxygen species. This dye changes from orange to green when free radicals are present. The free radicals are actually what slows and limits the cell death process by attacking important things in our cells like DNA, proteins, and lipids. Even though they seem very close in the amount, it's significant because of how reactive these molecules are. Lastly, we see the lipid or fat accumulation in the zebrafish from the red staining. In the experimental group, there was a less intensity due to free radicals that actually inhibit cell metabolism. When proteins are damaged, fats cannot properly function and complete processes in our cells. Fats are a really important and healthy part of our cells' function. The effect of the nanoplastic on zebrafish showed a relationship or correlation to cancer risk. When something is correlated, that means there is a relationship between two things, but doesn't mean that one always causes the other. That would be causation. If similar cellular changes occur in human, as the way they did in zebrafish, it could indicate a cancer risk for current and future generations, even if plastic production is banned today. In a 2022 study by Wang et al. Titled Microplastic Exposure Promotes the Proliferation of Skin Cancer Cells, but Inhibits the Growth of Normal Skin Cells by Regulating the Inflammatory Process. In this study, Wang and colleagues conducted an in vitro study to determine the negative impact of microplastics on both healthy and cancerous skin cells. In vitro studies are when testing takes place in a culture dish or test tube. The study used normal and cancer skin cell cultures that were treated with various concentrations of microplastic under different conditions. The cells were then analyzed and compared to detect the uptake of microplastics and determine any changes that might occur in the health and growth of the cells. The study found that both cancerous and non-cancerous skin cells had changes in their normal function and metabolism. This figure represents data collected by the researchers regarding the how of the damage. By using a spectrophotometer, they were able to observe light absorption in different molecules. I want to highlight the findings on NLRP3, a protein that regulates the immune system's response to harmful substances. When the skin cells were exposed to the microplastics, it caused the cells to produce too many harmful molecules, also known as free radicals. They were able to damage the mitochondria, which caused the membrane of the mitochondria to break, which led to the opening of certain channels that allowed damaged DNA to escape. This is what activated the immune system response from NLRP3. You may recall that the zebrafish from the earlier study also had inflammation from the uptake of plastic. In summary, the microplastics slowed the reproduction of the healthy skin cells and actually aided in a more rapid growth of cancer skin cells. What does this mean? This study suggests that high levels of micro and nanoplastics would most likely worsen any existing cancer. By creating an environment that is welcoming to tumor cell growth, this does occur with certain dosing and timing that we should keep in mind. A study done by Kathy et al., published in the Journal of Exposure Science and Environmental Epidemiology, investigates the link between cancer and concentrations of harmful chemicals like phenols, parabens, and PFOs. This was observed in participants previously diagnosed with cancer. When a study is done on data previously collected, it is called a cross-sectional study. After analyzing the participants' exposure to environmental chemicals found in their blood and urine, they were able to see if concentrations relate to cancer. During the study, they noticed that the female participants had particularly high elevations, more so than the male participants. Because of this, they focused in more on the female participants to see if there was anything else going on. They found was that environmental chemicals like PFAs, which are found in plastic, food, and beverage packaging, had a higher increase of elevated levels in the mostly female participants. The concentration and types of cancer varied among female participants based on their race. This serves as further proof that environmental factors and genetics play important roles in risk. The in vitro studies and in vivo studies are useful for understanding potential mechanisms and biological responses, but they may not fully represent how chemicals behave in the human body. This is because of the dosing amount that they use, or amount that humans would be exposed to day to day. So it's hard to directly apply the findings. A hazard is something that can potentially cause harm to you, like micro and nanoplastic. If you're around something a lot, you have a high exposure to it. So when you're thinking about your risk with plastics, you have to think about hazard times exposure. The Food and Drug Administration is in charge of regulating the safety of food manufacturing in the U.S. Since 2019, they have been studying per- and polyfluoroalkyl substances, PFAs. PFAs are found in products to stop grease, water, and heat and were developed in the 1940s. In July of 2020, announced voluntarily phase out by industry certain use of PFAS in food packaging. This decision was based on in vivo studies on rodents for a particular type of contaminant in the PFAs called fluorotelomere alcohol. They will continue to advise and enforce discontinuation of PFAs that end up being found toxic as research continues. February of this year, 2024, FDA announced that the PFAs used in the grease-proofing agents for food packaging were no longer being sold in the U.S. This is a win. What should we do and what should we believe when there's research findings that are showing a correlation, but the FDA hasn't decided about the specific regulations? We can decide to reduce our exposure to micro and nanoplastics while we await scientists' research decisions that truly give us the answers we're looking for. The few things that you can do: use your own water bottle instead of a single-use bottle, which has been shown and agreed upon by the FDA to have amounts of microplastics. If you do need to buy or use a single-use bottle, try to keep it out of the sun, as they are affected by UV light. If you're using a microwave, try to use glass instead of plastic to reduce leaching. There are also lots of types of reusable containers like stainless steel and even bamboo. We can work together within our different roles to keep everyone healthy and educated. Together we can ask our politicians, the FDA, and other important folks about what they know and what they think they're going to do about the nano and microplastic pollution that's harming our environment, as well as the lack of agreement and standardization on research practices regarding micro and nanoplastic, which have led to knowledge gaps among scientists, as well as different research findings between scientists on the correlation. To work on the knowledge gaps, scientists need to agree upon sample collection and preparation techniques so that the real-life mixtures of the types of plastic consumed from ingestion of food and beverages are being tested. Additionally, standardized detection methods should be agreed upon to measure the plastics from start to finish within the research. This also helps us understand and rule out other possible reasons for growth that give us the wrong information or the confounding factors, such as high-fat diet or smoking, which also leads to cancer. Then they'd be able to compare their findings. Without updates in these areas, knowledge gaps will continue to be there, and they will be unable to compare their findings to truly prove about micro and nanoplastic have a strong correlation to cancer.